Bismillah ar-Rahman ar-Rahim. This lecture is Introduction to Epidemiology. My name is Dr. Sireen Al-Khaldi. In this semester, I am going to teach you a lot about epidemiology as a science. Now, examining the history of epidemiology. There were 7 landmarks in the history of epidemiology that was started with Hippocrates. Hippocrates, who lived around 460 BC, found after his observation for human health and diseases, he concluded that environment and human behaviors affects health. Then followed by John Grant. John Grant in 1662, he quantified births and deaths. He started making lists and counting births and deaths of diseases. Third, we have Lind. Lind in 1747 found that scurvy could be treated with fresh fruit. Now we know that scurvy is caused by what? By deficiency of vitamin C. Right. That information was not available at the time of Lind. But from very critical observation for people who got scurvy and how to treat them and who does not have scurvy, he found that those with scurvy, if they eat fresh fruit, they get better. So we can see from the older days that the epidemiological way of thinking was able to solve health problems even without knowing the scientific basis and what caused the disease. Fourth, we have William Farr. This is more in the modern time. William Farr was in 1839. He established the application of vital statistics, making statistics for births and deaths and diseases, and he has started using that for evaluation of health problems. And he was in the UK or in London. Next came John Snow. John Snow in 1854, he tested a hypothesis on the origin of an epidemic of cholera that happened in London in that year. We will look at his story and what he did just in minutes. You should never, after this story, forget John Snow and what he did. Following John Snow, in the late 1800s, Alexander Lewis systematized the application of numerical thinking and quantitative reasoning and doing more analysis to statistics related to health. And finally, the new era of epidemiology started with Bradford Hill. Bradford Hill in 1937 suggested very important criteria for establishing causation, criteria that we apply to a relationship between a risk factor and a disease can establish the connection between the factor and the disease and may indicate causality. Now let's see the story of John Snow. John Snow was an English physician and modern-day father of epidemiology. He used scientific methods to identify the cause of the epidemic of cholera that happened in London in the year 1854. John Snow conducted a series of investigations in London. He conducted his classical study in 1854. Please remember this year, when in that year an epidemic of cholera developed in the Golden Square area in London. During the time of microscope development, or we can say before the microscope was developed and used, Snow conducted studies about cholera outbreaks, both to discover what caused cholera and how to prevent its recurrence. During that time, he used to argue and disagree with another scientist, William Farr. Remember, we talked about William Farr? They disagreed about what caused cholera. During that period of time, what was used to explain the causation of disease was the miasmatic theory. And this is what Farr adhered to and used to think about. Miasmatic theory says that there is like a miasma or a cloud with bad odor or smell that's near the surface of Earth. When people come into contact with miasma, they get the disease. This is how they explained contagious or infectious disease transmission at that time. So John Snow, from his previous experience in his work as a physician and seeing lots of cholera patients over the past years, he has developed an idea about water being the cause of cholera or such a disease. But of course, he did not know how would cholera be transmitted by water. And nobody even believed that because it was a strange idea. So what he did to start his study, he brought this map of the area in London where most of the infections clustered or accumulated, and started to plot the cases of cholera on the map. Each small black dot represents a case of cholera. And since he has this suspicion about water being the source or the cause of cholera, he plotted also the water pumps where people used to pump water and take to their homes every day. These are the locations of water pumps, the black circles with P in the middle that you can see in this map. When he looked at the distribution of the cases in these places and the water pumps, he discovered that most of the cases are clustering around the water pump in Broad Street that we can see in the middle of this map. So he became more sure or more confident that there was something in that water, but could not know what would that be. Since all that happened before the time of the microscope, they had no knowledge about what we know now as microorganisms, that there are small living organisms that cannot be seen with the bare eyes, but now we can see by a microscope. They are alive, they multiply, and they can be in water or food, and they can cause disease if we ingest them. They could not see this picture at that time. So he started to think that his theory has a lot of truth in it. So what did John Snow do? As we see in this picture, he decided to break the handle of that water pump so that people cannot anymore take any water out of it. People at that time used to come and fill the containers with water from that pump by pushing the handle. He broke that handle. And what surprisingly happened is that after removing the pump handle was removed on September 8th. As we can see in this figure, this is the Jon Snow epidemic curve. He drew this curve between the number of cases for every day during that outbreak, and after removal of the pump, the disease started to disappear and the cases were reduced dramatically. Now people started to believe him and to believe that there is something in that water that caused the disease. Now scientists started to believe his theory, and the authorities even believed that, and they made announcements for people in that area, announcement that we can see in this slide. This is the old printed one. On the right here we can see the photo of Jon Snow, and on the left this is the announcement to all people from the Board of Works responsible for water distribution. They are telling people here, you can make it bigger and read it all later on, but it's telling people not to drink any water that has not been previously boiled, and bring fresh water every day, boil it every day, and use it after boiling, and cover it whenever it's not used so that it would stay clean, and discard any water that remains at the end of the day, and the next day in the morning boil new water to use.
So Sir Austin Bradford Hill came up with this question. In what circumstances can we pass from an observed association to a verdict of causation? Upon what basis should we proceed to do that? To go from association into judging a causal association. So he came up with his criteria. Guidelines for judging whether an association is causal. So Sir Austin Bradford Hill came up with his criteria. First, we have these three most important criteria that should apply to a relationship between a risk factor and association to be able to judge that this association may be causal. The most important are temporality, strength of the association, and consistency. Temporality, as we have learned before, means that the cause or risk factor precedes the effect or comes before the effect. This should be confirmed. Second, strength of association. This can be known by a large relative risk. The larger the relative risk, the stronger is the association. This is a factor that should always be present. The third criterion is consistency. This means repeatedly observed by different persons in different places, different circumstances, and different times. They all observed such an association between this risk factor and the disease by different researchers in different countries and different research circumstances, and they all found an association. This is the third important criterion that should apply to judge that the association could be causal. There are some additional supportive criteria that if more of them applies to the relationship between risk factor and the disease, that could give extra supportive evidence towards the causal association. There is biologic gradient or dose-response relationship. If a larger exposure to cause associated with higher rate of disease, this means there is biological gradient. And if reduction in exposure is followed by lower rate of a disease, this is reversibility. This could support a causal association. There is also biological plausibility. This means according to the current knowledge that we have, according to the state of science that we know, the relationship between the risk factor and the disease makes sense according to the biologic knowledge of this time. Of course, this criterion is not obligatory to be present because our current knowledge about the biology is limited and other things may be discovered in the future. So it is not a must. Also, there is experimental evidence. If experimental studies have provided more evidence about this risk factor and disease, this would be a supportive criterion. There are also some other criteria like analogy. This means a cause and effect relationship already established for a similar exposure and the disease, and the one under study is similar to them. There is also specificity. This one cause is specifically leads to this one disease or effect. This could also be a supportive criterion. And the last is coherence. Coherent means that does not seriously conflict with the generally known facts of the natural history of and biology of the disease. It does go along and does not conflict with the known science in biology and disease occurrence. So the first three were obligatory to be there to judge for association, and the rest of them are there to provide additional supportive evidence towards a causal association. There is a very well known or the most famous cohort study in the world called Framingham Heart Study. This is an assignment for you to look for the answers of these questions on which an exam question will be in the final. Look for what is the Framingham study, when did it start and where, what was the disease studied, what are the most important findings, how many people participated, and how many generations it covered, and when did it end? So for how long did it go? Please go and search for it. You can find lots of information. It is a very well-known study.
One other important epidemic case study that we have to learn about is the London smog disaster that happened in 1952. Air pollution causes respiratory illnesses and death, as we know. When fog and soot from coal burning created a dense smog in winter of 1952 in London, this smog was around for 5 days, from December 5th to 10th of that year. There was a substantial increase in mortality. In the previous week, death rate or number of deaths in London was 2,000 cases of death. In this week of smog, deaths increased to 4,700 without any other explained reason. This is the epidemic curve that they prepared to study the reason for those deaths. We can see the number of deaths on the vertical axis with days of December of that year on the horizontal axis. When they plotted the number of cases of death with the increase in the level of smoke and increase of levels of sulfur dioxide in the air in London during that week, and also plotted the excess number of deaths during that same week, the connection was very clear. A sudden, sharp increase in the number of deaths was associated at the same time with sudden increase in smog that resulted from sulfur dioxide and smoke interaction during that week. The last epidemic case we will examine here is the epidemiology and polio vaccine. In April 1955, Dr. Thomas Francis, director of Poliomyelitis Vaccine Evaluation Center at the University of Michigan, announced that the 2-year field trial of the Salk vaccine against polio was up to 90% effective. That was a real cornerstone in prevention of disease for human beings. The results announced by Francis effectively marked the beginning of a new era and the end of polio as the most life-threatening and debilitating public health threat to children all over the world. In this slide, we can see the Legionnaire's disease outbreak. What happened in that very famous outbreak? Members of the American Legion gathered for the annual American Legion Convention held on between July 21st through July 24th in 1967 in Philadelphia in the United States. Soon after the convention began, a substantial number of attendees were admitted to hospital emergency departments or were examined in doctors' offices with acute onsets of fever, chills, headache, malaise, dry cough, and muscle pain. The more troublesome is that during July 26th to August 1st, that's after the convention ended and everybody went home, a total of 18 conventioneers who were sick, they deteriorated and died, reportedly from severe pneumonia. On the morning of August 2nd, a nurse at a veterans hospital in Philadelphia called the CDC, that's the Centers for Disease Control and Prevention. She reported cases of severe respiratory illness among convention attendees. Later that day, subsequent conversations with the public health officials uncovered an additional 71 cases among persons who had attended the convention. The goal here was to find out why these conventioneers were becoming ill and in some cases even dying. Now we can see the epidemic curve for Legionnaire's disease epidemic. Here, this is the number of cases by day of occurrence or reporting during July and August in 1976. We can see the peak of cases between 25th to 27th, just after the convention ended. This may indicate the latency period. People got infected during the convention and got the disease clinically after that. And following August 2nd and 3rd, the number of cases decreased dramatically. Here we can describe who got the disease. We have the age groups of persons who got the disease, and also the where, the places where attendees of the convention attended. A group were in Hotel A, another group in Hotel B, a third group in Hotel C. When we look at absolute numbers of cases, it is not always very informative when we analyze data for an epidemic. We always need to calculate a rate. So here we can see, for example, for each age group, there is the total number of those in that age group and the number who got ill, so that we can calculate a rate of disease in that age group, or rate of disease in that certain hotel. Okay. After looking at the previous slide and calculating the percentage of those who developed the disease, that's what we call the rate of the disease among all different hotels, A, B, and C. We found that those who stayed in Hotel A had the highest percentage of illness. It was 9% in Hotel A compared to 6.8 and 5.4 in other hotels. When we looked at the different age groups, we also found that the age group that's over 70 years of age had the highest rate of disease, highest percentage among all age groups, which was calculated by dividing the number who got sick divided by the number of people, total number of people in that age group. We saw also the rate of disease was 20.4 in that age group, and it was much lower in all other age groups. So by calculating those rates, we got deeper into analyzing who got the disease and where. From there, we can make inference. We can infer that there is a connection between staying in Hotel A and becoming ill. And we can also infer that older persons are somehow more susceptible to the disease. 5 months after the first cases of Legionnaire's disease occurred, results of the case control study indicated that spending time in the lobby of Hotel A was a risk factor for illness. In January of the next year, 1977, the Legionella bacterium was finally identified and isolated and was found where? It was found to be breeding in the cooling tower of the hotel's air conditioning system. The bacteria then spread through the building whenever the AC system was used. Similar bacteria grew in warm water in nature, such as hot springs, and also has been identified in air conditioning cooling towers. The finding from this outbreak investigation led to development of new regulations worldwide for air conditioning systems. And from here, the name Legionnaire's disease was a new name of a disease that refers to this severe respiratory infection that happened to those who attended the American Legion Convention